Welcome, everybody, to this new episode of CCIR's Student Spotlight. With me today is Amy Thomas. Amy is a current junior at Valley Christian High School in California. During her time at CCIR, Amy took the course on cell and developmental biology at the Future Scholars Program with Dr. Mamiko Yajima from Brown University. For her final project at CCIR, Amy decided to focus on oncology and in particular looking at some issues related to metabolism and genetics. It's a pleasure to have you here today, Amy.
Thank you. So I'm so glad to be here. Again, my name is Amy, and the main project I did this time was mainly on non-coding RNA's role on cell differentiation, and especially the area of circular RNA on how metabolism happens in oncology and cancer cells and how we can mainly control that. So what I really explored in the beginning was the Warburg effect, which is actually a relatively unexplored region that goes into cancer metabolism, where instead of, let's say, regular ATP generation, with glycolysis and the citric acid cycle that goes into oxidation phosphorylation, cancer cells mainly only rely on glycolysis. And so that means that a lot of the genes that goes into regulating cancer cells are going to be targeting this Warburg effect. And so the main area of non-coding RNA I studied was circular RNA, which has been known to overexpress in these cancer cells by actually regulating another regulating RNA, which is called miRNA.
Fantastic. I'm glad we can get straight into talking about your research topic. So one thing I want to talk a little bit about, I guess it'd be useful for the listeners maybe first to get a little quick overview of what exactly is cell and developmental biology. So what, broadly speaking, were you guys looking at in your particular course with Dr. Yajima?
Yeah, of course. So generally, cell and developmental biology is just looking at cell differentiation, like saying how when the cell really first starts, with the use of different protein coding RNAs and also the ones that help with regulating factors, it's really saying where the cell is going to end up, after all these little components differentiate them into the final component. And that's why things like stem cells are so heavily researched, because we're just trying to do a lot of genetic manipulation and trying to study how the cell gets to where it is.
And I suppose when you are looking at oncology in particular from this sort of developmental biology perspective, what you're kind of looking at, I'm guessing, I'm not a scientist, is more of how that process of cell replication and so on goes wrong. Is that sort of what you were specifically looking at?
Yeah, so that's pretty close to what it was. So mine is mainly studying like what happens when the cancer cell already has formed. So it's really not preventing the cancer in the first place, because that is a very hard thing to do, but more like regulating it so that we make sure the cancer cells that are already present doesn't continuously generate energy, for it to keep replicating.
Interesting. So I want to quickly ask how it is that you, I guess, first chose this particular course. What was it that drew you to become interested in cell and developmental biology?
Yeah, sure. So mainly, I've always knew that I was kind of like a science kid. I like doing labs, but mainly it was in AP biology where we started learning about all these non-protein coding RNAs, so like the regulating ones. And because we didn't cover too much about it, but I also found it really interesting, I decided to just want to do some outside research. So I went to CCIR, dove into a little bit more specifics about the non-coding RNA, and that's how I kind of ended up here, because it turns out a lot of these RNAs that I was looking at, they really target oncology. specifically.
So you mentioned AP biology there as sort of your gateway to getting more in depth about biology. I'm curious to hear a little bit about how your experience with Dr. Yajima and doing this sort of research-based course where you're actually conducting a project, focus on something relatively specific rather than an overview of all of biology, the way that AP works. What has been sort of your experience with these two courses? What are some of the similarities and differences? And in particular, what were some of the challenges when you transitioned to, let's say, this higher level and more intensive course that we offer here at CCIR?
Yeah, of course. So mainly my experience with CCIR has been pretty good. So Miss Yajima was super nice and super helpful because she kind of understood that this was like one of our first times, leaving our traditional academic curriculum, to really researching something that at least we felt like we were interested about. So she was really nice. She was very knowledgeable, in her own area. So in metabolism. So whenever we, mention our interests in what we want to do, our topics, she was always super helpful with trying to help us narrow in what we wanted to do, and also along the way teach us how to do these research and how to actually word and how to write these essays, because they are a little bit different from our traditional, like even academic essays. They do deviate a little bit about that. And so actually along with that, our TA, Mr. Dang, he was also super helpful in that because he kind of taught us how the research process is, how to find good resources versus a little bit more biased and unreliable sources. So with that, it just really helped us with our scientific, I think, development, like the scientific process. So mainly through CCIR, it was me gaining the experience of learning how to research. And outside of that, they also gave me a lot of support in detailed corrections with my essays, things that may seem too generalized, too broad, all of those things. And because it was like a newer experience for me, since I never really got outside of my curriculum to do independent research like that, my biggest struggle was probably getting started with the essays, choosing a really specific topic to write about because there are just so many out there, and really getting started with that. But I feel like through Miss Yajima and Mr. Dang, they have really helped me, kickstart this project and still give me that passion to continue exploring it in the future.
So you talk a little bit here about the differences in writing a research paper and your traditional academic essay. I wonder very briefly, what are maybe one or 2 points of like sharp difference that you notice between these two ways of writing?
Yeah, of course. So with academic essays, we would usually just give like a really stern assumption about something. Like we keep making statements about why things are. And, usually in order to write an informative or argumentative essay, that's what you do. We try to convince someone of something. But with these essays, it's very informative, meaning that we don't want to make concrete statements, because everything that goes into research are things that are still, in progress of studying. So everything is not really, I guess, concrete in that way. So whenever we write, we try to be informative, being like, well, that's what we've seen these days. That's what we can assume, but we can't say this is what's going to happen because this is an area that's still being researched.
Interesting. This connects to another question that I thought of when you were talking, which was, how has this experience, so you talk about yourself as a science kid coming into the program. How has this experience, I guess, changed your understanding of what science amounts to and what the real scientific process actually is?
Yeah, so in the beginning, I kind of felt like I was a science kid mainly because of the content we learned. So I always felt like that was super interesting. So I just wanted to learn more content, if that made sense. But now I think it kind of changed my view because instead of just, continuously learning, I wanted to apply that knowledge to real life situations. So let's say we learned about the non-coding RNAs in AP Bio. Okay, but then now I was able to apply that knowledge and actually research into how it affects cancer cells, what we can do about it, you know, maybe presenting it as like a future target um, gene that we can use to treat cancer patients.
Hmm.
So it's really the shift in mindset, yeah, for me.
Right. So from more of a theoretical, let's say, to more of like a hands-on and engaged sort of perspective. Um, Excellent. So I'm curious also, so you talked about how the most difficult thing for you in the whole research process was actually picking your topic, which, by the way, will be true at the high school level. It will even be true at the PhD level. So if you actually end up sort of embarking on this journey, you'll learn that this is the problem that never goes away. Um, and then eventually you'll have too many topics that you're interested in. And it's not that you don't know what question to ask, but you just have too many questions to ask. Um, but I'm curious how it was that you eventually settled on this particular topic that you ended up working on.
Yeah, so between just like the broad categories that I was interested in, so like I knew I wanted to do genetics, the application of genetics, but also I wasn't very interested in developmental, as in like stem cells and what it goes out to. So I knew that it wasn't going to be like a regulatory, like in stem cells, like a regulatory process in stem cells. So I kind of did narrow my way towards, cancer cells and how circular RNA is going to impact that. So just RNA in general. And the more I looked into it, I knew I wanted to do something that's a little bit more, I think, niche than just what we learned. So let's say the microRNAs, I didn't really want to do that because we already learned some basics, in AP Bio. So I just wanted to explore something that was like completely new to me, but still in the field that was
Mm.
like super interesting. So I guess I was just trying to explore new things that I didn't know before, and also tying it back to some of the common things that we know, like, oh, cancer grows. So it's mainly looking into like the little components that people may not always think about that contributes actually a lot into the overall bigger issue of cancer.
Mhm. So you started the our conversation off by giving us a quick overview of what exactly it was that you did. And I wonder if at this stage you can say a little bit more about sort of the um, you were gesturing at it a little bit, but the potential sort of concrete applications of this particular research or the downstream interests from a medical perspective of what you were looking at.
Yeah, sure. So from the research, it was mainly we gathered that generally circular RNAs are always overexpressed in a lot of cancerous cells because they actually inhibit other like good regulatory RNA sequences. So what scientists have really been, I think, pursuing is to track these high levels of RNA of circular RNAs that are expressed in cancer cells in hopes of having a therapeutic target, meaning that if they notice that, hey, this area has a lot of circular RNA cells, maybe that's saying that there's a lot of cancer in this area. So mainly for targeting maybe early developing cancers. So exploring that a little bit, in addition to how we can actually modify the circular RNA so that it becomes something like, instead of inhibiting good RNA regulatory genes, it could actually inhibit bad RNA regulatory genes, so that we just kind of shift its purpose a little bit to help with the overall like oncology study.
And as you see the state of research today, how distal is this particular application in the future? Do you estimate, or do you have any sense of? I know scientists are relatively uncomfortable making these sorts of predictions, but do you have a sense of how the sort of how how close we are to these sorts of technologies and so on being impactful in our daily medical lives?
Yeah, sure. So I know that there has been, a lot of scientific discoveries all the time. So right now, I think even though this is like a very, I think, likely solution, or at least something that will help with the cancer treatment, I don't think it's going to be as focused on as, let's say, bigger issues. Like, let's say the current issue of, actually, no, not the current issue, but the current development of like CRISPR. So the bigger, more common genetic technologies, that's probably going to be what's more focused on, because I know that there have been a lot of efforts everywhere on, trying to stop cancer. So I feel like this one, out of, let's say, thousands that are out there, it may not have a primary priority over everything else. But
Sure.
for sure, I think over time, as little bits of technology and advancement come up, eventually this will be pushed up the list, and it will be something that will help with the cancer treatment.
Um, I'm also interested because you keep talking about making these tiny, tiny contributions. I think a common theme when I talk to a lot of students who work in STEM and do research for the first time is how it shifts their understanding of how science actually functions and progresses. Um, because in our sort of scientific textbooks, in the way we learn science, it's often going to be sort of big names. So you hear Newton, and then you hear about Einstein, you hear about Max Planck, and whatever. And it's going to be these sort of great figures, great scientists that are pushing the field forward. As students sort of actually get into scientific practice, what they realize is that science is much more like I don't know, a hive of bees than it is sort of these majestic geniuses. Um, Majestic geniuses exist, obviously, and they're very important to science. But how has this has this at all shifted your understanding of what your aspirations as a scientist might be, how you think of the field, and how you see the value of this practice?
Mhm. Yeah, like you said, we do hear a lot of these big names. So obviously we assume like, hey, these are the people that actually, let's say, like did something or invented something major that was like super impacting. But yeah, over time when I learned a little bit more about biology, researching, especially now that I was doing, this primary research paper, I did feel like I realized that a lot of actually advancements are through collaboration, and it was mainly that we build off of, let's say, another person's work, and we, in that way, we kind of do like, we kind of just collaborate by building on top of knowledge. So maybe it takes that one person, to make that big discovery that really links all the studies together and really puts them together into a functional, let's say, technology. But for sure, I think I realized that science was more collaborative than I expected. So that probably means that in the future, I I would probably consider like scientific research and to be a scientific researcher as like one of my career options for sure. Like my one of the top priority career options I have, and it's mainly just because it's been really helpful getting to learn everything and being able to apply that with hands-on research. So in that way, I realized that it wasn't just me going out to do scientific research to be that big name, that pops up, but mainly to contribute to like an overall movement to help with the scientific advancement.
That's fantastic to hear. With that, I think I'm going to move into what is typically my final question for students on the Spotlight series. And the question is just this. So Amy, you have gone through the 13 weeks of CCIR where we give you a lot of work, make you do a lot of readings, where you do a whole bunch of writing and do something that maybe you've only sort of tried a little bit before, which is sort of intensive academic research. A lot of students coming into the program were like you, who have never done research before coming to CCIR, and they see this program and they are a little bit nervous and daunted by how challenging it might be. So for students who are interested in research, but a little bit daunted by it, I was wondering, Amy, as someone who's come through this process, is there anything you would want to say to them in terms of advice, insight, warnings of how best to sort of satisfy this interest of theirs?
Yeah, sure. So I feel like I felt kind of the same way, like all the students you mentioned. I was a little bit, I think, intimidated as well. Like, oh, we're going to do actual research. Like that was kind of a little bit of like a leap of faith that I had to do, in order to do this research and actually get going with the research project and actually coming out with a final result. So honestly, my biggest advice is to really just take that leap of faith over, your fears, because ultimately it's like you can't really fail, and because either way you're able to gain knowledge, you're able to make something that's your own project, and whether or not you feel satisfied with it, it's still like a foundation that you can build off of, and it could actually motivate you to do future research and to improve on your work. So mainly I feel like we shouldn't view this as like a scary challenge, but it's mainly like something like a catalyst for you to grow in your knowledge and to actually enable you to explore your interests. So I think to utilize it as a resource, not like a scary platform that is really that you have to reach a certain like expectation to. So it's all really just a part of the learning process.
That's a great answer. I think that's one of the better answers I've ever heard. You cannot fail. That's a great piece of advice, because even in failing, you are learning and growing, and so on. I think that's absolutely true. A lot of, especially the students that come to CCIR, put a lot of pressure on themselves. They're very excellent students. They're trying to go to the best universities, do research as a high school student, and they end up putting too much pressure on themselves, and they forget that, as you were saying, Amy, even when they fail and they don't manage to write the research paper that they hope for, they've learned so much along the way and grown so much along the way. So with that, Amy, I want to say it's been absolutely so interesting and wonderful to learn about your experience here at CCIR, about your work, about your journey. Your insights for the other students have been also wonderful. And so with that, I want to say thank you so much, Amy, for your time today. And I wish you all the best of luck in all your future scientific endeavors.
Yeah, of course. Thank you for having me. It's been great being here. So I'm just happy to, be a part and being able to share a little bit about my journey.